Well, thanks for everyone for joining. See a lot of friendly names and faces, and appreciate everyone joining. We're at a critical time in the COVID-19 crisis, and my colleague Edgar Davidson gave a talk about COVID-19 at the very beginning of this in March and April. We're entering a new year and really a new kind of era in the development of the COVID-19 vaccines and therapeutics. And as scientists that work on COVID-19 and other viruses, we're constantly following these things and are pleased to be able to share our perspective with everyone. The perspective I'm going to present today is from myself, but from a scientist perspective. I'm not a medical doctor. I do have a PhD, and I have studied virology and viruses for the past 25 years. But I certainly can't comment on some of the medical parts of things and clinical parts of things, but I can tell you a lot about the virus and about the vaccines that are being developed and how we're dealing with them at Integral Molecular. One of the key questions, of course, everyone asks is really, when's my life going to get back to normal? And I think we have a pretty good sense of when that's going to happen these days with the vaccines starting to come out. So we'll try to answer that question and how to get back to normal and survive the next 6 months. As a little bit of background, I run this company, Integral Molecular. We're at 3711 Market Street, so just next door to Venture Cafe, which is now in this space that's blank in this picture. But we've been here for actually the Science Center for about 20 years. We're founded by virologists, myself and Sharon Willis and Joe Rucker, many others now. In the past 2 decades, we've been focusing on manufacturing enveloped VLPs, virus-like particles, which we call lipoparticles. We sell them commercially. We manufacture reporter viruses. We've published over 50 manuscripts on viruses, and we've worked on all kinds of viruses. Of course, now SARS-CoV-2, HIV, dengue, Zika, Ebola, Marburg, chikungunya virus, influenza, some of the encephalitis viruses, hepatitis C virus, to name a few. So when the coronavirus outbreak really started this past year, this is something that we're heavily involved in now, but we've always been working on outbreaks of viruses, the outbreaks of Ebola, the outbreaks of Zika virus, the outbreaks of chikungunya virus before that. We're very involved, usually with the NIH and National Institute of Allergy and Infectious Diseases, in understanding those outbreaks, combating those outbreaks, and ultimately helping develop vaccines and therapeutics against those viruses. So when coronavirus broke out, of course, we were very involved and got involved, and we still are very involved in that fight. Now, I'll describe some of what we're doing a little bit later on. Before I go into what we're doing and what's happening with vaccines, just a little bit of a primer about what the coronaviruses are and just a little bit of history to put it all in perspective. So coronaviruses, electron micrograph pictured here in the middle, are a family of these enveloped, what's called positive sense single-stranded RNA viruses. So they have this RNA genome inside of them. They have an envelope, a lipid membrane on the outside of them. And then, of course, they have these spike proteins, pictured in purple here, that are responsible for getting the virus into the cell. And these viruses are named because of the crown-like structure on the virus, although I don't know if it looks much like a crown to me, but it looks like a crown to someone who discovered it, and they called it coronaviruses. And up until about 2002, there were many coronaviruses known, but they only caused kind of cold-like symptoms, nothing significant or very serious for the most part. And then, of course, in the past 2 decades, there's been 3 coronaviruses that have emerged. The first one in 2002 was SARS, what we call SARS-1 now, the original SARS, which started in China, transmitted from civet cats, this beast over here, again in a live animal market. It infected about 8,000 people, caused several hundred deaths, but it kind of fizzled out. And It's been well studied since then, but it really hasn't emerged again, at least the SARS-1 since then. The next outbreak was in 2012 with a related virus called MERS, Middle Eastern Respiratory Syndrome virus. It started in Saudi Arabia, transmitted from camels to humans, infected several thousand people, and had a pretty high fatality rate, actually. It actually is still circulating, although it's not very high, but it's still around. And then, of course, in 2019, the end of 2019, what happened was a variant of the coronaviruses related to SARS evolved, and it became what we know now as SARS-CoV-2, which is the actual virus name. The disease, just to make sure everyone's got the same nomenclature, the disease itself is called COVID-19. The virus is called SARS-CoV-2, which is not easy to pronounce or write, but that's the official name of it. And of course, it started in Wuhan, China, likely transmitted from bats, possibly through pangolins, which is this creature down here, which is really cute in a strange way. And of course, it's not cute when it's gotten to over 33 million people and 1 million deaths. Actually, this is outdated now. It's beyond that, and sorry to say, but the fatality rate is about 3%. So it could be worse, but that's still obviously a cause for alarm and has caused a worldwide shutdown. So one of the first questions we asked as scientists was, where did SARS-CoV-2 come from? And when you look at the sequences of the virus, the actual DNA or RNA, in this case, sequence of the virus, and you trace it back to other coronaviruses and what their sequences look like, it's pretty clear that SARS-CoV-2 came from another bat coronavirus. It might have gone through pangolins at one point to get to humans, but it's pretty clear that it came from bats pretty close to that. One thing that is also very clear is it was not man-made. There was some speculation at the beginning, and certainly there's other speculation out there and conspiracy theories that it was man-made, but there's really no evidence for that. The sequences and cells were traced back to other bat coronaviruses and pangolin coronaviruses that existed well before 2019. The other part to think about for COVID-19 is how transmissible is it? Of course, we've heard a lot about this in the news, that it's kind of like the flu, that it's gonna, that it's not that transmissible. And we didn't understand a lot about COVID-19 or SARS-CoV-2 at the beginning, but we understand a lot now. There's been a lot of research, a lot of scientific research about how it's spread. The good news about COVID-19 and the virus is that it could be worse. It's definitely very transmissible. And of course, it's spread mostly through respiratory droplets and aerosols. But you can see this comparison here. This scale here is a measure of how transmissible these viruses are. And COVID-19 is high, in comparison to many of these viruses, on the higher side of things. But you kind of see this other scale down here with measles, which is highly contagious, and we're nowhere near that kind of contagion level. So despite all the chaos and disruption this year, the good news is that it wasn't as bad as it could have been if the virus was more easily transmissible. The other thing from a scientific standpoint was when this first emerged was, could this have been prevented or predicted? Viruses emerge all the time. Ebola, Zika, where did these things come from? And the unfortunate part is that the viruses are out there. They're always out there. They're out there to kill us. And they're spreading because we travel, and because birds fly, and because insects migrate or get around and travel on our suitcases when we go from place to place, and airplanes and cargo containers. And the world is an international place, and viruses tag along with insects and animals and with us as we travel from place to place. And so this emergence of a new virus is surprising on one sense, but kind of predictable on the other hand, that something would emerge in all of these interactions. And it's really highlighted here. There's a paper published by a number of colleagues of ours, actually in 2015. And you can kind of see the title, a SARS-like coronavirus is poised for human emergence. And this is 4 or 5 years before COVID-19 hit. And it's based on the propensity of this kind of virus to mutate and to evolve and to infect humans even better than the original SARS coronavirus. So while viruses have been around for centuries, the emergence of new viruses that are trying to attack us is unfortunately not unpredictable. And more viruses will emerge in the future, unfortunately, but that doesn't mean that we can't be prepared. That's what science is about. That's what public health is about. That's what being prepared is about. Matter of fact, when we'll get to this, a lot of the reason that we've been able as a community to develop a vaccine and drugs against this virus so quickly, in less than a year, is because of all of the preparedness that has gone in and the investment that's gone into science and medicine and global health prior to the actual pandemic. So just a little bit of terminology before we get a little bit deeper into the virus. I'm not going to go too much into the technologies or the virology behind things, but just some basic terminology that everyone should be aware of. This is a schematic of the virus, and this protein on the outside, the spike protein, which sometimes called S protein, is something that probably most people have heard of at this point. And when you look at the structure of that protein, shown here in red and blue, there's a domain of the protein called the receptor binding domain, RBD here, that's responsible for exactly what it's saying, binding a receptor, the ACE2 receptor to be specific, which is on the cell surface. So this is how the virus gets into your cells. It has all of these spike proteins on the surface of the virus. It binds to an ACE2 receptor, which is on the surface of many of your cells. The virus binds, and after it binds, it actually undergoes a whole transformation of its structure, and that's kind of shown here, that allows the virus to actually fuse into the cell and deliver its RNA that then produces more viruses in the cell. So this spike protein, and in particular the receptor binding domain, are particularly important to understand because those are the pieces of the virus that are the major target for vaccines, for natural immunity, when you develop natural immunity after you're infected, and also for many antibody therapeutics that are being developed. They're all, for the most part, focused on the spike protein, and almost all of them to date are focused on the receptor binding domain, this critical part of the protein that's necessary for its function. So as everyone's, I'm sure, familiar, there's many different vaccines in development and now approved against coronavirus. And I'm not going to go into all of them, but I will point out that there's many different flavors of these things. Traditionally, a traditional vaccine used to be either a protein or essentially a killed virus or an attenuated virus, a weakened virus. And those are still being developed, but they take longer to develop. They're more expensive to develop, and but they're really well tested and very safe. So proteins are being developed by Sanofi GSK, and different viruses, either attenuated viruses, or actually they're using another virus like adenovirus to deliver a piece of the coronavirus, are being developed by AstraZeneca, which is actually now partially approved, and Merck and J&J, Janssen. I should mention, a company in our own area actually started at the Science Center even, in Novio, and the Wistar Institute just a few blocks away are developing a DNA vaccine against coronavirus, and they developed this similar product against SARS-1 that's in, I believe, in clinical trials. So these new vaccines are based on DNA or, of course, RNA, which is related to the DNA, a different kind of nucleic acid. And these are much faster to develop and much easier generally to manufacture. They're relatively new, but the technology is evolving very quickly to be able to use these and develop them and get them into people, as we've seen, in less than a year. The interesting thing about this, I think, especially being where we are, is that, and I don't know if everyone's familiar with this, but of course there's two RNA vaccines, one from Moderna, the other one developed by BioNTech and partnering with Pfizer. But those two RNA vaccines, which are the main vaccines on the market in the US right now, were all stem from technology that was developed at Penn, University of Pennsylvania, a couple blocks away, by a collaborator of ours, Drew Weissman, and one of his colleagues there, who, a decade ago optimized the composition of the RNA to make it immunogenic, or actually to reduce immunogenicity, to make it immunogenic against the coding part of the RNA. So I won't go into all the details, but that innovation, which is possibly Nobel Prize worthy, really enabled the RNA vaccines that we're seeing today. There's a decade of work, and that got licensed to Moderna, got licensed to BioNTech, and they partnered it with Pfizer. All comes out of research that was done just a few blocks away from us. I think also, just to point out something that has been in the news quite a bit, that there are many vaccines in development. At one point, I don't know if there still are, but there was 170 different vaccines being developed, and for good reason. This is a public health emergency, a worldwide pandemic, and so many different approaches and people and companies and laboratories trying to fight this pandemic. At one point, there are 26 of these in human trials, which is really unprecedented. And almost all of these, if not all of them, focus on developing antibodies against this spike protein, this external protein of the virus that's essential for its function. To put this in perspective in terms of the development time, this is an article, it was actually published last year by, I believe, the New York Times. Even they overestimated how fast or how long it would take to develop a vaccine. So in yellow here is the normal vaccine development time. If you started at 2020, a normal vaccine would take 10 to 15, maybe even 20 years to develop and involve months and years of clinical trials. And to put that in perspective, everyone knows at this point the mRNA vaccines have been developed in less than a year, and actually other vaccines too are starting to come on the market and developed in less than a year. That is lightning speed. For science, we've never seen anything move that fast. And again, to put that in perspective, you look at this table on the right. For a typical virus, or in the past at least, the cause of a disease has taken typically months, sometimes years, to figure out. For SARS-CoV-2, it really took days to figure that out. And to sequence it, to get the actual sequence of that virus, took days, not months. What was really surprising, although, well, unprecedented certainly, is that the structure of that protein came out 3 weeks after the sequence was published, after the sequence is available. Typically, that takes years. For something like HIV, for instance, the sequence came out in the mid-'80s, and the structure of the equivalent protein, the envelope protein for HIV, came out about 10 years later. Here it took 3 weeks. It's just mind-boggling how quickly things have gone. The first therapeutic antibodies took 3 months to discover and get into trials, 4 months to get into human trials, vaccine trials. 4 months, and therapeutic antibodies and vaccines in less than a year. And typically that's taken a decade or longer to get. So from a scientific perspective, no one has ever seen science and the clinicians involved move this fast, which is really encouraging. It's never as fast as you want because we're all dealing with the consequences of this pandemic and still taking all these precautions and not living the normal life that we were used to. But it's still unprecedented how quickly things have gone. Again, just to put this in perspective too, it was possible at the beginning of the pandemic that no vaccine could have been discovered. You know, if you look at something like HIV, we've been working on HIV and HIV vaccines in particular for more than 35 years, and we still don't have a vaccine. We're really still far from it, in my opinion. We've still got a ways to go. And that's partly because the HIV genome replicates very quickly and has a high mutation rate. So it's taken decades to do. For influenza, again, it's been around for decades. Vaccine development for influenza has gone on for decades. We have vaccines, but of course, as you know, we have to get a new vaccine every year for influenza. There's no universal vaccine that protects us from all of the different strains and serotypes of influenza. And that's because influenza has a kind of a unique genome. It's called a segmented genome, which means it has different pieces and they can recombine. And that makes the virus very agile. It can evade the previous year's vaccine and means you need a new vaccine. So as scientists, this is what we're working on. How do we develop vaccines against very difficult viruses that mutate around the vaccines like HIV? How do we deal with viruses that are really a whole subset of viruses underneath that, like influenza? And you know, again, for perspective, the smallpox vaccine almost 100 years ago, it's great when it, you know, that was the first one developed in, you know, the thousands of years of humanity, but it's only 100 or so years old. We're still dealing with a relatively new age of vaccines, but when you have it, it can result in complete eradication. So in the news these days, of course, are mutations of the virus, and we're starting to see that. And it is kind of inevitable that SARS would mutate. You know, will SARS mutate? Of course it will. As virologists, we know that that's what viruses do. They change because they replicate so quickly. There are thousands, millions, sometimes billions of viruses in a given infected person, and each of those viruses usually has small mutations. Many of those mutations kill the virus and don't have any effect. Many of those mutations don't do anything. But if you've got so many mutations floating around, at some point you get one or two or more that actually make a difference and allow the virus to evolve and become more infectious. And that first came up really for SARS-CoV-2 with this D614G mutation. And this is the structure of spike protein. The mutation D614G is right here in this little blue amino acid pictured here. And you can kind of, you see how quickly these things can spread. The D614G is in blue in these maps. And before March 1st, and it was very localized in Europe. And after, less than a month later, it was everywhere. And really it was the dominant form of globally of SARS-CoV-2 very quickly. You know, of course, now in the news, we're hearing about more variants. And to be honest, there likely will be more variants coming out as the virus continues to replicate. That's just what viruses do. There's more mutations. And if they have some kind of advantage in spreading, they're more infectious, they replicate faster, they interact with the receptor better, whatever it is, that gives each virus an advantage and it begins spreading after that. So there's two variants in the news right now. One is the UK variant that many people have heard of. It actually has 17 mutations. It says formally the name is WIV1-CoV and has 17 mutations in blue spread all throughout the spike protein. And those mutations appear to allow this virus to spread more rapidly. The good news is that not every mutation allows the virus to escape. That is, just because it has a mutation doesn't mean it can evade all of the vaccines and drugs that are being developed. And we'll get to that in a second. As a matter of fact, for the UK variant, there's really very little evidence that it can escape vaccines, but it is a concern. It's something that as scientists, we have to watch. And you can kind of see here highlighted in red in particular, this is the spike protein interacting with the ACE2 receptor. So this one mutation in particular likely enables the spike protein to bind much better to the ACE2 receptor, which allows the virus to spread and infect better. A newer variant is this South African variant, 501V2, which again has many mutations spread throughout the spike protein, has several close to the ACE2 binding site. And this one has more of a concern for vaccine resistance based on where the mutations are localized. I think most of this will still be fine, but it's definitely something we have to test for to make sure that the vaccines, the drugs that we're developing now, are all protective, even as the virus continues to evolve. So this is the main question that we're often dealing with these days: is will the virus evade these vaccines? We're spending a year developing these vaccines. If a new variant comes out quick enough that can completely evade the vaccine, that's a pretty significant event. But that's not likely to happen, fortunately. And there's several reasons that the virus is not likely to evade the vaccines or the drugs, actually, the therapeutic antibodies at least, that are being developed. So the first reason is that the spike protein actually has several sites of vulnerability. So all of these sites, for instance, are places where antibodies, your part of your protective immune response, can bind to the spike protein and kill the protein and ultimately kill the virus. So it's not just the vaccine doesn't always doesn't usually produce just one type of antibody. Most vaccines can produce many types of antibodies, and there are many types of antibodies out there that can protect you, that can kill the virus. So the fact that this protein, this virus has many sites of vulnerability, allows vaccines to work in multiple ways. And even if a virus mutates to avoid one of these, it's unlikely to mutate to avoid all of these sites of vulnerability. The other factor involved in making this unlikely that the virus is going to evade the vaccine is that the virus itself, this particular virus, doesn't seem to mutate particularly rapidly. So if you look at the sequence variation of all the different kinds of strains out there, and you look at the data, which is essentially what's pictured here in terms of where variation occurs, it doesn't really mutate all that much. It does mutate. There's always this low-level background of mutation, but it's not, the virus doesn't replicate as fast or make as many errors every time it replicates. So it's not a huge mutation frequency. So it's likely that the virus won't be able to mutate faster than we can immunize. That's a guess. We have to see how it all plays out, but it's pretty likely that the virus will be killed by each of these strains, each of these evolving strains will be killed by the vaccines that are currently under development. Actually, the third part of this is that the vaccines that are being developed appear to be pretty robust. This is some of the data, and I have some more on the next slide, but the vaccines in humans and animals are producing highly neutralizing antibodies. These antibodies kill the virus, and they kill the virus in multiple ways, these multiple sites of vulnerability. So the fact that we're getting good immune responses, and we're seeing these 95% plus protection rates in humans is a good sign that the vaccines are very effective, that they can keep up and protect us even as the virus continues to evolve. The related question with with the vaccines is how long are they going to last? And we don't know completely the answer to that question yet. But again, the good news and the promising news, which I'm very hopeful for, is that these vaccines are going to last probably years. I don't know if they're going to last forever. That is, you get one shot and you're protected for life. It may require a new immunization, a new vaccine shot every 5 years, 10 years. We just don't know yet. But it's likely that the vaccine is going to work for certainly at least months and probably years. And we know that from several lines of evidence at this point. So partly it's from natural infection. If you look on the left here, you can see what survivors of infection look like. Even after 4 months after natural infection, they still have high levels of protection with their antibodies. And that seems to be persistent. It's going to go down eventually, but at least after 4 months, and again, probably much longer. Even more encouraging, there's recent data, this just came out last week, from Moderna, where their vaccines in humans are lasting, they published data for up to 4 months. And you can see for most of these patients, the protection seems to be persistent and not going down too much, although it depends what assay you use. These are binding assays up here. These are neutralization assays down here. And the neutralizing antibodies do seem to be going down in some patient populations. These are older people, these are mid, and these are younger people. So your immune system will lose protection over time, but it looks like the vaccines are going to be highly protective. They're going to last at least 4 months and most likely several years before everyone needs a new shot. But that's certainly going to be long enough for us to conquer this virus, get rid of this virus, and get back to a more normal life, which is encouraging. So hang in there, get your vaccine shot when it's available, and we'll all be protected. The other thing that gives us a little bit of information about how long the vaccine is going to last is that if you look at SARS-1, the first SARS that broke out in 2002, the natural antibodies from that outbreak lasted for about 1 to 3 years, depending on the patient. And after a few years, the immunity, the natural immunity to that virus declined. So if the vaccines are similar, if natural infection of SARS-2 is similar, we can expect somewhere between 1 and 3 years. And that should be long enough to eliminate the virus and at least eliminate it from pandemic status, even if there are localized outbreaks. So one of the most important things, at least from my perspective, in the science about coronavirus has been how do you get understanding how you get infected. Certainly, as a virus company, as a virology-based company, we're asking this question all the time. How do you get infected? Because we don't want to get infected. And the bottom line is you get infected mostly through coughing and sneezing. If you look at these rates, it's estimated that you need about 1,000 virus particles to get infected. And that's because some virus particles are dead, some virus particles are not going to replicate quick enough to really start an infection. Some are going to be killed by our immune system. But if you get about 1,000 virus particles, you're probably going to be infected. So to get 1,000 virus particles through normal breathing, you have to be in the presence of someone else breathing relatively normally for about 50 minutes, almost an hour, because they shed about 20 viral particles per minute in normal breathing. If they're talking, that rate goes down by 10. So within 5 minutes, 200 viral particles per minute, you can get 1,000 particles and be infected. And when someone coughs or sneezes, they're releasing about 200 million viral particles out of their nose or mouth, essentially instantaneously. And if you're within that radius of where they're coughing or sneezing, you've got a pretty high chance of being infected. So this is the main way of transmission. There are certainly other potential mechanisms of transmission, but this seems to be the primary means of transmission. And probably more important than understanding how people are infected is how you can avoid being infected, of course. So there's some really interesting and really phenomenal studies at this point, scientific studies. Some of them are case studies, but really good analysis of how people have become infected from SARS-CoV-2, especially early in the pandemic when not everyone was wearing masks and taking precautions. This is one example here on the left, a call center where everyone in blue on this side of the building got infected, or at least half the people got infected. The other half of this floor in this building, hardly anyone got infected. And that's because people are sitting in proximity, breathing the same air. This is a really interesting study on the right, where there's a small restaurant in China, filled with patrons, and this person in yellow here was the source of infection. Everyone in red also got infected, but there's a few really interesting things to note. First of all, the air conditioner for this restaurant was here, blowing this way and then recirculating the air this way. And you can see that everyone in that pathway had a high chance of getting infected. Interestingly, not everyone, right? Only about half the people did get infected, but none of the people that were outside of the airflow of that air conditioner got infected. So how do you avoid getting infected? Social distance, stay apart from people, and stay in fresh air, which means stay outside, increase the airflow if you can have fresh air coming through, open a window, use an air purifier, but stay in fresh air, and of course, wear masks. And masks are a really important part of this because we can't always control our environment or the people next to us. So there's another really good scientific study about mask use that came out in May, where 2 hairstylists in Missouri ended up testing positive for COVID. But before they stopped treating their customers, they actually treated 139 clients while they were COVID positive and symptomatic. They were coughing and sneezing. Don't ask me why they were treating, but they were. But they wore masks the entire time. And as a result, not a single client, none of those 139 people tested positive. None of them caught COVID from those hairstylists. But the family members of those hairstylists at home, where they weren't wearing masks, did contract COVID. So masks completely work. They absolutely work. But it's really important to understand, again, from a scientific perspective, not all masks are the same. This down here is a mask. It does not work for protecting you from COVID. It just covers your eyes. This over here is an N100 respirator. It's like a HEPA filter over your mouth and nose. It's about $35 on Amazon. It works. It will filter everything that's coming into your mouth and nose for breathing. And everything in between. There's surgical masks, there's N95 masks, the bandanas and the other type of filters, they work, but they're really not as good, especially thin cotton masks. Some of them are very poorly protective. Just because you're wearing something over your mouth, a thin cotton cloth, doesn't mean it's going to be protective. Similarly, if a mask has a one-way exhalation valve, they kind of look like this. Actually, this one has it too, so you have to be careful. It protects you because it's filtering air going in, but it's not protecting other people around you. So you have to wear something over it in addition, like a surgical mask, to protect other people. So keep in mind, wear a mask, but make sure you're wearing the right mask to protect you and to protect others, and make sure you're wearing it correctly. If air is escaping, around the mask, or you're wearing it over your chin, it doesn't work, right? You got to wear it over your mouth and nose with a tight seal to make it work effectively. And this over here is some of the data, the graphs of showing how well some of these masks can filter versus some, something like a thin bandana mask, which hardly filters at all. Then the question is, can all these precautions stop the virus? And the answer is unequivocally yes. These have been really phenomenal tools to help us prevent infection across the world. And if everyone was doing it, we'd have a lot less coronavirus right now. And we know this because we can track what's happened with other respiratory viruses. So this is the flu season in the southern hemisphere this past summer. So summer for us, winter for them. And you can see the normal flu season, which is kind of in orange here. It starts, it peaks, it goes down for all of these southern hemisphere countries. In 2020, in the darker shade, the red, it started in February, March. And then as soon as everyone started clamping down, keeping social distancing and masks, it was completely eliminated. In Australia, New Zealand, Chile, Argentina, South Africa, you name it, flu season was virtually eliminated in 2020. So we can stop a respiratory virus if everyone is taking the right precautions. Which kind of gets me to what we're doing at Integral Molecular. Just give a little bit of background. We start, as I said, we've been working on other viruses for 2 decades, and a decade before that, at Penn and other research institutions. We've worked on HIV and dengue and Zika and chikungunya. When coronavirus came out, we started turning our attention and our tools and our resources to helping in that fight. So we've made reporter viruses for serum testing. So we make these reporter viruses that are safe, they're quantitative, they're kind of used as a critical reagent for testing patient samples for their response to the virus. So for instance, if you're running a clinical trial and you're testing 1,000 patients or 10,000 patients, and you need to know if your vaccine is protecting those patients, inducing protective antibodies in those patients, you can get serum from those patients, run it through these screens, and find and quantify how protective those patients, quantify the antibodies in those patients' serum. So that's actually being done quite a lot these days with our reporter viruses. We do epitope mapping, figuring out where antibodies bind on the spike protein. So the spike protein footprint in blue is kind of where it sits, and then these particular residues, the red and red, are where the energetically critical residues of how the antibody binds to the spike protein. And those are particularly important because those are the residues that actually determine most the resistance of the virus to antibodies and therapeutics and vaccines, which is another thing that we do over here, engineering the spike protein for testing for resistance, particularly to predict resistance. And then we also test for specificity. We have a technology called the membrane proteome array that tests against all 6,000 membrane proteins in the human genome. And we test antibodies all the time for this, for specificity, before they go into preclinical trials and INDs that are filed at the FDA. And actually about 25% of the antibodies that we test are polyspecific. They bind more than what they're supposed to. And this is actually a real example of an antibody against SARS-CoV-2 that also binds another human membrane protein, completely unrelated, but could cause significant adverse effects in those patients if they receive this antibody. The thing I'm certainly proud of as a company is that we've actually been able to offer many of these technologies for free this year. We along with everyone else, are very affected by the COVID shutdowns and pandemic, and we want to do our part. So we've actually been able to send our reporter viruses and epitope mapping and some of our other technologies, make them available for free to over 100 COVID laboratories around the world. As a company, we're human and we're affected by this as well. And as virologists, we talk about how we can protect ourselves. So we've taken ourselves 35 different actions to keep everyone safe in the company. We're still operational. We've been mostly operational this whole year. supporting other COVID laboratories, as well as other pharmaceutical companies and laboratories that use our services and products. But we've done it in a safe way. And you can operate in this COVID environment and do it safely if you take the right precautions. So they kind of break down to these different categories: social kind of changes, physical changes, PPE, protective equipment changes, monitoring, and knowledge of how the virus spreads and how to protect yourself. All of these things, which of course includes social distancing and masks and controlling airflow and air purifiers. We bubble wrap the entire office. It looks like it's been attacked by bubble wrap. We don't travel. We don't have visitors coming in and out. We do Zoom meetings like everyone else. We work on shifts as much as possible. We screen temperatures. You know, some things are obvious and some things maybe a little bit more subtle, but I think it is possible to operate in a COVID environment in a safe way. And that's been very important to us as a company. I think one of the other things that's been really important for us this past year, which is kind of COVID related in a weird way, is the disparity in social inequity in our society, which really has been heightened, and certainly the awareness of it has been heightened by COVID. The fact of the matter is that COVID-19 has disproportionately affected Black and other minority communities, both financially and health-wise. And as a company that is in some way benefiting from the COVID-19 crisis, in the sense that we do COVID research and we help companies fight COVID-19 and other laboratories, this is our job. But the fact that it's disproportionately affecting minority communities is something that's not lost on us. So what I'm really proud of in our company is that we've taken these things side by side, and that we've implemented 30 anti-racism initiatives, things that we can do. Some of these are still in process. Some of these we've been doing actually for years. It's just enhancing them even more these days. Focused on education, on jobs and training, financial aspects of what we can do to support minority communities, mentoring, social and corporate initiatives, all things that we can do and we have been doing for years, but even more now, and really even more important for everyone to be involved in these anti-racism initiatives. Again, connected in a strange way to COVID-19, but really directly for us. We feel that it's our responsibility and our duty to bring this to take action, not just because of the social responsibility, but because of the scientific responsibility as well. The two are interconnected. And then finally, one of the other things that we do as a company, and again, we feel it's our responsibility, is to communicate science. So certainly here, to make sure that we're talking about science and explaining what we know to everyone, but also in other ways. Not everyone is a scientist, not everyone understands science, and not everyone is going to read the scientific articles that we work on. And we realized this, many years ago, and that many people are interested in science but don't understand it, or maybe biotechnology is a black box. And it's our responsibility in part to explain that to everyone. What do we do? What do scientists do? So a few years ago, we started a bioart residency program, actually with Angela McQuillan at the Science Center and the Esther Klein Gallery. We put together a bioart program where we've invited artists to come into our laboratories, spend 3 months with us at our lab meetings, talking with scientists, using our equipment, using our reagents and supplies, whatever they need to express what they're doing, to understand, to communicate science in a different way. The mission of the program is really to improve that understanding of science and biotechnology in all of our lives, to create this dialogue between artists and scientists, certainly happens within our company, and then ultimately to have this positive impact on human health. And we've seen many examples of this along the way. We've had 8 artists to date. Actually, we have our next one starting on Friday, tomorrow, and Rebecca Rutstein. And this piece here was actually created by Laura Splann, who did her artist in residency remotely this past year during the pandemic. This is actually a movie, a visual of SARS-CoV-2 spike protein, which she imaged using some of the software that we use in the lab to image proteins. And she made it into really a phenomenal movie. There's if you search her name, and this is some of the information here. It's called Disrupted Domains by Laura Splan. You can find this on the web and see the movie in action. It's really phenomenal. So when is my life going to get back to normal? With everything that we understand, at least in my opinion, as a scientist that studies this, I think we're going to start seeing normal, some kind of normal, really start in the middle of 2021 as vaccines roll out. The faster those vaccines can roll out, and depending on how many people actually take the vaccine, that's another issue we won't talk about today unless people are interested. But the faster everyone can get immunized, the more we can all get back to some kind of normal life. And I think that's going to happen in the late spring, summertime, at least that seems to be the track now. I think it's also important to point out, though, that normal is going to be different. And it's going to be different, especially for the rest of the world. What I'm talking about normal here is Philadelphia and probably most of the United States and many other developed nations. But we've got 8 billion people on the planet, and not all of them are going to get vaccinated by the summer. And it's likely that many of them may never get vaccinated, or it's going to be many years before they get vaccinated. So the rest of the world may still be experiencing the SARS-CoV-2, the coronavirus 19 outbreak for many more years, which affects all of us directly or indirectly, and we should be aware of. So I left plenty of time for questions, hopefully, so I'm happy to answer anything. Our sponsorship here, we do get funding from NIAID, the NIH. Thank you to the Pennsylvania Department of Community Economic Development, DCED, who's given us funding for some of our COVID research, and Life Sciences PA, who's also been very supportive of our work. And thank you to everyone at Integral Molecular who's made all of this possible. Great. Thank you, and happy to take any questions if anyone's interested.
Seeing a lot of clapping emojis, which is awesome. A lot of thank yous in the chat.
So Ben, this is Bob Bowdish. My question is, with the nature of the virus and what is being communicated in the public, I have concerns that we're going to have a higher, group of people who are unwilling to take the vaccine. And what concerns me is it will probably be less educated people. And now we end up, back where we kind of started, right? Like the people who are probably the most needed, most lack of education, et cetera. I hope I'm coming across, in the right way. How do we, what I try to do is anyone that I talk to is, discuss the science and discuss how the clinical trials are conducted and the manner in which they're conducted and all that. But do you have concerns, or are you hearing anything that is being done to try to give people the education so that they can buy into taking a vaccine?
Yeah, it's a great question. It's a huge concern. It's frustrating, as someone who's worked on vaccines one way or another for, almost 30 years. You work so hard to develop these things. Obviously, billions of dollars have been spent, and all of a sudden someone says, I don't even believe it works. How do you convince those kinds of people that this is real? And of course, part of that is explaining science, which we try to do. Some of it's educating the public about the science and the medicine and the safety that goes into these trials, which is all true and accurate. I think that the component that is going to convince that segment of the population to get a vaccine is actually not going to come from the scientists. And I think that that's something that's kind of being overlooked right now. there's this level of trust that it's very hard to communicate facts and figures and statistics and clinical trials to people who won't listen. And I think the answer to that is that you have to communicate in a different way. And that's partly what our bioart residency is about. And I see Laura Splan on here is communicating some of that, and using these art forms, communicating to people in an emotional way, in a way that they, on their terms and their language, in a way that they'll understand and trust, from sources they'll trust. I think that's the way. I can't throw statistics and charts and graphs at someone and say, believe this. That's a hard thing to do for a certain part of the population. So I think it's got to come from education in different ways, but it's going to be a huge factor. And I don't know if we're spending enough time talking about that or doing enough on the education front yet. We're not that.
I think you're right. I think they need to find the right people to communicate it. They need to figure out, it's basically a marketing job, right? You have to identify who your target market is and then go get the people. But it is concerning to me because I think it will be a bigger problem than we expected. When you're looking, what is it, 70% we're trying to get to? Yeah. And
or more.
Right. And what is it, 40% right now are unsure?
Yeah. I mean, I think we haven't, that's not the bottleneck right now, of course. We're still getting the vaccines rolled out, but you can see in a few months that may be the bottleneck, that not everyone wants to get it. And then there's this huge educational push that requires a lot of planning and preparation and thought and strategy to get the right message to the right people. That's something that we can potentially start thinking about and get a head start on. Ultimately, it should be the federal government and the NIH doing that, but sometimes that happens, sometimes that doesn't.
That and you have a group of people that are bound and determined not to believe what the government tells them or what NIH tells them or whatever. So, but thank you. I've been concerned about it, and it's good to hear I'm not going crazy.
Yeah. I think also, you see Tony Fauci on the news just about every day, and I think he's been very, very careful about his messaging, to talk as much as he can in the right terms about how this vaccine is being developed, how safe it is, how effective it is, the controls and experiments and rigor that goes into these experiments and these clinical trials that's not rushed. I think he's been very careful to spell that out. But, again, not everyone's going to take that at face value. They should. He's really a great scientist and done a great job leading this. But, there's a lot of work to convince people.
Yeah, I think the primary care physicians are going to help a lot. I mean, there's just not very many that don't believe in vaccination. So I think they'll help too. They're going to need to talk to their patients. But it's something for everyone, I think, to think about as they're talking to people. We have to make sure that people know that they're safe. And if you're suggesting to someone that they shouldn't take a vaccine, you might be actually doing them harm and someone else harm by doing so.
Yeah. I think there's a question. I don't know if I lost you. Vinu, is that?
Hey, Ben, how are you? Happy New Year to you.
Hey, good to see you.
Hey, likewise. Quick 2 questions. I guess we are almost out of time now. Any thoughts on these variants that we are seeing from South Africa and UK? Will the current vaccines be effective or not? As you mentioned about Tony Fauci, I mean, nobody knows yet, but do you have any thoughts or any insights? And the second thing is, like we're talking about this delay in, these vaccines getting administered, right? So for them, as of now, like only 4 million were administered till on 31st of December. So, and when you hear from the doctors, they say there's we have to wait, there's a lot of paperwork to be filled, then we have to wait for 30 minutes for the side effects and whatnot. Any insights on how can this be improved or, these are full questions, but would love to hear what you have to say about that.
Yeah, well, so the variants, as I mentioned, there's many variants coming out. That's what viruses do. They keep evolving. But the good news is that it looks like, and it's very likely that the vaccines will be able to effectively kill all of those variants. There's still data to come out and really prove that, but the virus doesn't mutate fast enough or far enough usually to completely evade these kinds of vaccines. And there's enough sites of vulnerability on the spike protein that the vaccine should induce a very broad neutralizing antibody response. So I think we'll be okay as long as the vaccines roll out quick enough and the virus doesn't get a head start. It doesn't mean there can't be a variant in the future that evades the vaccine, but I think we'll be able to eradicate the current SARS-CoV-2 using the current vaccines. In terms of the rollout, what was the question exactly? Like
I mean, any
dose?
thoughts on the deal? Because we are still only 4 million people that were vaccinated by the end of the year, right? And when I have a few colleagues and friends who are doctors who are administering these things, and they are not very happy because it's still a long process. It takes like an hour just to get one person done from beginning to end, right? So
that's not quite a scientific question. It's really a logistics question, and that it requires a lot of coordination and a lot of planning to get those things done in an effective way, which clearly has not been done as well as it could have. The vaccines themselves they're kind of, at this point, they are what they are for right now. They're not going to really change in the immediate future. Maybe longer term, they'll do some other trials and figure out if they can dose them a different way or store them a different way, or reduce the side effects where those are coming from. But I think probably for the next 6 months, those are the parameters we're going to have to live with these current vaccines. They're not perfect, but they're pretty good. And certainly in terms of protection, they're very good. Just the logistics of getting them into everyone is not perfect. So we're going to have to live with that and build the infrastructure around it to make sure we can deal with it. But that requires a lot of coordinated effort, which is sometimes not always happening.
Yep. Thanks.
I know we're a little bit over time, Ben, we really appreciate your time. There's one last question in the chat.
Oh yeah. Do you have any concern for antibody-dependent enhancement? Yeah, so ADE, antibody-dependent enhancement, is actually quite a significant problem for dengue virus, where you have these different serotypes of the virus. And if you get one serotype, you get sick, but you recover. If you get a second serotype, however, you have antibodies that are kind of residual from the first serotype that actually make you sicker when you get a second infection. So that's kind of what this refers to. And you can have those antibodies for many different reasons, from other SARS or coronaviruses, or from a vaccine even, or from a prior infection. There's some evidence for SARS-CoV-2 that ADE, this enhancement, could be an issue, but I don't think it really will be at the end of the day. It is for dengue, but that's because dengue has these very defined serotypes, and they circulate at different points in time in different locations and in different years. For coronavirus, you've got all of these strains mixed up, and you've got pretty much this, there's variants, but they're all kind of the same serotype. There's the antibodies that neutralize one strain are still going to neutralize the other strains for the most part. So I don't think ADE is going to be too much of a concern for coronavirus, but we have to watch it. It's something that we have to watch for with the vaccine development, with therapeutic antibodies, and with patients that recover. But it is something to watch.
All right. Well, we're 5 minutes over, so Ben, we really appreciate you sticking around. I know people have a million questions, but we appreciate you tackling the ones we've presented tonight. So thank you everyone for tuning into this session. There will be an artist studio tour at 6:30, which we highly recommend you attend. If not, enjoy the rest of your evening, and we hope to see you again next week.
Okay, thanks everyone.